Many areas of science use serial dilutions in preparations for different experiments. Serial dilutions are often used in the fields of microbiology, biotechnology, and in chemistry, just to name a few. Therefore, a clear and concise approach to the learning of this important concept is essential. Serial dilutions are usually made in increments of 1,000, 100, or 10. Today we'll be working with 10. The concentration of the original solution and the desired concentration will determine how great the dilutions need to be and how many dilutions are required. Important also is the total volume of solution needed. If only small quantities of solutions are needed, then greater numbers of dilutions will be necessary. The most common examples deal with concentration of cells or organisms, or the concentration of a solute. Using serial dilutions instead of making one big dilution in order to finally arrive at the desired concentration is not only cost effective, but also allows for small aliquots to be diluted instead of unnecessarily large quantities of materials. For this lab exercise, we'll find out the concentration of an undiluted sample of E. coli in conjunction with the spread plate method of inoculation for counting microbes. Let's begin. So as you can see, you're going to start with an unknown concentration of E. coli. In other words, there are E. coli cells in this liquid sample, in this liquid suspension, but we do not know how many there are. We have 4 blank serial dilutions prepared, labeled A, B, C, and D. Tube A will be dilute to 1 to the 10. Tube B would be diluted to 1 to the 100. Tube C would be diluted to 1 to 1,000, and tube D would be diluted to 1 to 10,000. Once you have your tubes marked, it's also a good idea to mark your pipettes. We're not going to do a very serious scientific version of a serial dilution, but instead this will give you a general idea of the broad concept of serial dilutions. So, the first thing we need to do is transfer 0.5 milliliters of the unknown concentration into tube A. Each of the tubes A, B, C, and D contain 4.5 milliliters of sterile water. Therefore, 0.5 milliliters being transferred from the unknown sample into tube A would result in a 1 to 10 dilution. Therefore, each time we take that dilution and transfer 0.5 mil to the consecutive tube, we multiply by a factor of 10. So I'm going to reach all the way to the bottom. Before I take the sample, it's a good idea to draw up several times a full pipette of the liquid and extrude it back into the tube. This helps ensure that it is mixed properly. I'm going to pull up approximately 0.5 mLs. That's approximately halfway up my pipette. I'm not going to insert that into the liquid of tube A, but instead I'll hold it above the tube. Next, I'll use the pipette that I labeled for tube A to mix again my sample. Once I've done that a couple of times, I draw up again approximately 1/2 mil, put that into tube B. Notice each time, guys, I'm using a different pipette. I don't want the concentration of what's inside the tube that I'm drawing from to contaminate the tube I'm going to. So again, approximately 1/2 mil after I've mixed up a couple of times. Transfer that to tube C. Now I'll use tube C's pipette to do the same thing one more time, and we will go finally into tube D. We now have a 1 to 10,000 dilution factor. So from this point, we're ready to plate our sample. I have 2 plates prepared. One says 1 to 10,000, one says 1 to 100,000. In order to plate these samples, I'm going to use the same pipette, since tube D is the same concentration. The difference here is the volume in which I will pipette to the 1 in 10,000 plate, and the volume I will pipette to the 1 in 100,000 plate. The 1 in 10,000 plate would get approximately 1 milliliter. Since that is a 1-to-1 dilution factor, you can see 1 in 10,000 tube, which is tube D, would also be the 1 in 10,000 plate. For the 1 in 100,000 plate, I need to reduce that by another factor of 10. So instead of 1 mil, I'm going to use 0.1 mil. I'll draw up approximately 0.1 mil right about there. Placing 0.1 mil on the 1 in 100,000 plate will give me the desired result. Next, we're going to use the spread plate method, and we're going to spread those cells around the plate very evenly as possible. Make sure you touch the sides with your L-shaped spreader and go all around the plate several times. L-shaped spreaders are, of course, a biohazard and should be disposed of in the biohazard non-sharp. I'm going to use a separate L-shaped spreader for my next plate because I have two separate concentrations. Again, once I have spread those around evenly onto the plate, I'll dispose of this in the biohazard non-sharps. Okay guys, so let's look at the results. On day 2, in order to help count the colonies, you may have to do something like this. Notice how many colonies are on this plate. So I've divided the plate into 1/4 of a section. I'll count the number of colonies inside here. At this point, I've determined it to be 75 colonies. I'll multiply that number by 4 and come up with 300 colonies for my plate count. This plate count is also viable, and I'll explain why in just a moment, but I can count these individually on the plate. I don't need any help. I've determined there to be 30 colonies on that plate. There are two ways that you can tell if you have a viable serial dilution. When you're conducting a standard plate count, the ideal number of CFUs, or colony forming units, should be between 30 and 300. This is the window of accuracy we are looking for in this experiment. Also, if there are around 10 times as many colonies on the 1 in 10,000 plate as there are on the 1 in 100,000 plate, your serial dilution was accurate. Now let's get to the math. So as we stated before, we have our undiluted sample and we have our 4 tubes. Tube A is 1 to the 10, 1 to 100 is tube B, 1 to 1,000 tube C, 1 to 10,000 is tube D, and we have our 2 plates, our 1 in 10,000 plate and our 1 to 100,000 plate. So again, keep in mind the volumes that we're working with here are 4.5 milliliters in each tube. So if we start out by drawing 0.5 milliliters from our first tube, our undiluted sample, placing 0.5 milliliters in 4.5 milliliters is a factor of 1 to 10. We would then continue that process, pulling out 0.5 mil from each tube until we reach tube D. At that point, we would pull out 1 mil, which is a 1-to-1 dilution factor, 1-to-1 dilution factor, so that would be 1 to 10,000 dilution factor, the same as tube D. We'll pull out 0.1 milliliter from tube D onto our 1 in 100,000 plate. The 1 in 100,000 plate is the plate you will count. So let's say, for the sake of argument, remember we said for our purposes a number between 30 and 300 was going to be great. But for this demonstration, let's use 10. I count 10 colonies on my plate when I come back. In order to calculate the number of cells in the undiluted sample, I simply take that number and I multiply it by the inverted dilution factor, which is 1 in 100,000, which gives me 1 million colony forming units per milliliter in the undiluted sample.